Hello and welcome to our talk for poster number C359. My name is Trang Do and I'm a researcher in the Center for Bioinformatics in Saarland University, Germany. The title of our talk is Deregulation of Epigenetic Marks Associated with Differential Exon Usage of Developmental Genes. So let's start with getting familiar with the important term for the study. So first of all, the concept of exon usage is linked to alternative splicing, and alternative splicing processes mRNA from transcription by including or excluding exons in the mature mRNA, and thus create multiple variants from a DNA template. So this inclusion or exclusion lead to the change in exon usage and is the key of our talk. And we also know that there is local enrichment of histone mark around the splice sites from many previous studies, and so this is basically one aim of a study, that is to investigate the local effect of the histone mark at histone batteries that affect mRNA splicing. We also know that alternative splicing and histone modification has together found in cellular and systemic development. And the question is, now if we associate the differential histone mark with differential exon usage, we will see this connection shed light on cell reprogramming. And this is a question we aim to clarify with the help of 19 cells and tissues coming from the Human Epigenomes Atlas. This is a project belonging to the Roadmap Epigenomics program, and those include, for example, stem cell or cell of different lineage coming from mesoderm, endoderm, or ectoderm, and of different developmental stages. So for the 19 cells and tissues or samples, we use the RNA-seq data to study differential exon usage and ChIP-seq data to study differential histone modification for 5 different histone marks. For those differential analysis, we perform pairwise comparison across 19 samples. So after that, we need to perform multiple testing correction to reduce the bias. After that step, we associate DEU from differential exon analysis with differential histone modification M value, and we gather genes where the correlation between the two features are high and call them episplice genes. So those are the main steps of the workflow, and I will go more into detail now. So first of all, we define exon flanks, which are the region around the splice sites of an exon, and this is the region where histone modification would be annotated to that stretch around 200 base pair up and downstream from the splice site. After that, we perform differential exon usage DEU using DEXSEC and filter out the DEU event using the adjusted p-value of less than 0.05. Simultaneously, we perform differential histone modification with NA norm and also filter out the DHM events using PM value and the p-value. Since the number of pairwise comparisons was really large across 19 samples, we had to perform multiple testing correction. After that, we filter out the DEU that happened in more than 90% of the pairwise comparison. And in the last step, we eliminate the DHM that have more to do with transcription than splicing by eliminating those at the first exon of any gene. After revising the reliability of DEU and DHM analysis, we now link them together and look at the overlap or the correlation between the two features with, first of all, odds ratio and then Pearson correlation. And we filter out episplice genes on the basis of Pearson correlation using an absolute value of more than 0.05 and adjusted p-value of less than 0.05. To investigate the episplice genes from the previous step, we use target index, adjusted RAND index, and gene ontology enrichment analysis. Coming to the result, we first identify gene with DEU and DHM overlap by an odds ratio of more than 1 and Fisher exact test p-value of less than 0.05. While there is an abundance of gene with odds ratio more than 1, the Fisher exact test p-value is a very stringent filter and result in only a handful of significant genes shown in red. So that's why we look at the linear relationship between the features with a less stringent test, that is Pearson correlation. And we gather the episplice genes, which are again a gene with high Pearson correlation coefficient, and HGFR2 is in fact one instance of those genes. It is detected from the pair mesenchymal stem cell and sigmoid colon, where there is this diagram showing exon usage and the signal for A3K27MET3 for this gene. And in the DEU analysis, we see that sigmoid colon actually contained more of the B variant of FGFR2, why mesenchymal stem cell contain more of the C variant. Meanwhile, we see that the signal for A3K27B3 is higher in mesenchymal stem cells, suggesting that there is actually a anti-correlation between the DU and DHM in this case. We also found evidence with experiments showing the anti-correlation of the histone mark with the inclusion level of this exon 4 C variant in epithelial in mesenchymal transition. In the next step, we look at the pairwise distance between tissues using the shared episplicing genes and target index, and we found clear cluster of cells coming from early stage development, like H1 stem cell or endoderm, mesoderm, neuronal stem cell. We also look at the label for potency, type, and origin and life stage of those cells and compare that with the cluster coming from our target index distance and look at the matching between the two labels. We found high overlap between the labels, indicating that the episplicing genes was able to distinguish the cells based on those 4 criteria. In the last step, we performed gene ontology enrichment analysis for each of the histone mark or old episplicing genes together, and we saw a lot of terms coming out to belong in developmental processes like organization or cell growth. For the analysis of all episplicing together, we also see a lot of terms for organization and morphogenesis. So as a summary, we found that the differential incidence of protein isoform is often associated with histone mark change at the boundary region of an exon, and many of those episplicing coming from our study are associated with development and metabolism. So this is the end of the talk for poster C359. Thank you for your attention.